Good morning. My name is Carrie Brown Lima, and I'm the director of the New York Invasive Species Research Institute here at Cornell University, and we're going to be hosting this special webinar today. We've received a number of requests for an update on the research project to develop a biological control for water chestnut, and including recently one from the Fish and Wildlife Service and the Connecticut River watershed, as well as our partners here in New York State. So we're very grateful today that Dr. Bernd Blossey, who's an associate professor in the Department of Natural Resources here at Cornell University, has agreed to provide an overview of the research that's been done over the past over 17 years with collaborations from China and here in the US alike. And he'll be talking for about an hour, and we may go a little bit beyond 12 for those of you who are interested and have extra questions. However, we ask that everybody save your questions for the end. We do have all lines muted, and we can take questions via chat at the end, and we'll read them in the order that they come in. This webinar is being recorded, so for those of you who cannot participate in the whole webinar or would like to share it with your colleagues, it will be available later on this week on the New York Invasive Species Research Institute website, and we'll send the link to that through the chat box. So with that, I'd like to pass the stage to Dr. Blossey, who will get started with his presentation. Thank you all.
Thank you, Carrie, for the introduction. Welcome, everybody, to this after snowstorm Tuesday. I hope you all had a great time. I tried to, not always had to. And I will try to brighten up your day with a little bit of update on the biocontrol of water chestnut. I'm not in this alone. There's obviously a whole bunch of people that are working on it with me. In particular, Wade Simmons, who may join us in a little bit. He's in class right now, who's a graduate student doing most of the activities. He joined the team about 3 or 4 years ago. And then early on, it was a postdoc of mine, Jianqing Ding, who is back in China, who did much of the early work on water chestnut. It has taken this long for many different reasons, and if I can advance my screen, we will get to that. And I can't advance my screen. Oh, there. So here's a little bit of an overview of how this all developed. As Carrie said, we are in this for about 17 years now, although the work program really were 3 or 4 intensive years early on between 2002 and 2005, where we had some political funding through a New York congressman that gave us $250,000 and $250,000 to water milfoil work to work on aquatic nuisance species here in New York. That's when Dr. Ding joined our team. He came to Cornell as a postdoc. He was a well-established biocontrol worker, a scientist in China already at that time. So we had that expertise. We worked both here in New York and to some extent in China, where a bunch of tests were done. So what we did during that first 3-4 years was really look at whether there were natural enemies of water chestnut in New York, in the Finger Lakes regions. And those of you that have experience with water chestnut will know that there is actually the native water lily leaf beetle does attack water chestnut, but doesn't seem to have a control effect on water chestnut, despite the species being occasionally very abundant on it. A second work program then was look at Asian specialist herbivores as biocontrol agents, particularly in China. There was an early collection, literature review, and some field surveys by Bob Pemberton, who did some of that work in the 90s and maybe before that. So we had some kind of an idea of what organisms may be available. And we also tried to begin the development of a monitoring protocol that could be implemented before release of biocontrol agents. Would assume that was focused on other floating aquatic plants really at the time. So after we did quite a bit of work early on, and identified Galerucella birmanica as the most promising agent. And I will get into a little bit of the life history in a moment and review that. We looked at some host specificity. Dr. Ding tested quite a few plant species in China. There were some issues with getting North American plant species into China due to quarantine regulations, so that didn't work out really well. We couldn't even get North American water chestnut into China. They just didn't release them for tests in the field. So there's all kinds of logistical problems that were part of that early exercise, but mostly the funding dried up. So we couldn't do anything for 10 years until we received additional funding from the New York DEC through the New York Invasive Species Research Institute to restart the work on it. So we had a good foundation for the work, and we then began by early on drafting a full host specificity test plant list that was required by regulatory agencies. A little step back, those of you that may not know how weed biocontrol agents are regulated in the US, there's a review group called the Technical Advisory Group, which in USDA APHIS, who reviews petitions for test plants that need to be tested for particular organisms, reviews the data, makes a recommendation to USDA APHIS. That will then be reviewed by a federal agency, and it incorporates a Section 7 review by the U.S. Fish and Wildlife Service, and then you may get a federal approval for release, followed then by individual states who would need to approve the release of biocontrol agents. That may differ from state to state, depending on where you are. You may need to find out about how that works. So, but the initial step after you have an organism, you need to draft a host specificity test plant list that can be anywhere from 30 to 70 different test plants, depending on how isolated taxonomically the plant is. Some of you may have seen that list that we circulated, and we settled on some 56, 57, or 58 species. I think it's 57, that we then proceeded to test. We had some ideas of wanting to do this in China, but because of the problems of getting material of US plants into China, we settled on the need to do this work in quarantine. We are fortunate enough to have a quarantine here at the Cornell University, and so we received an import permit from the feds, from USDA APHIS, to allow us to bring Galerucella birmanica into quarantine. So that's where most of the work has been done that I will be talking about. So we then focused on host specificity work at Cornell and additional other ecological work that I will very, very briefly mention as we go on through this in the common garden and also in the field. A couple of years ago, a different morphological type was discovered, whether that's Trapa bispinosa, Nancy Rubicki with the USGS called it bispinosa. I looked a lot into the Chinese literature on different morphological forms of water chestnut. They treat them all the same because a lot of the morphology is based on the taxonomic differences are based on nut morphology, which can be very, very variable. There needs to be some genetic work that some of the Chinese are doing, some of the people here are doing. Nancy Rubicki is following up on that one. For now, I'm not inclined to call anything that we have in the US, despite different morphologies, anything different than Trapa natans. And our insects don't care either, so that's just upfront about that. We had hoped to complete the host specificity testing in 2018. I'm going to go into that one and submit the tag petition for review. That will likely, it didn't happen in 2018. We will try to do this next year. We'll prepare most of the materials. So we think we have a great organism here that we consider safe for release in the US to just get the punchline. And I will tell you some of the problems that we're encountering that's typical for work in quarantine and going through a weed biocontrol program. So that's basically the timeline here, how we went over the last 17 years to end where we are here today. So this is the critter that we are working with. It has multiple generations a year, similar to the water lily leaf beetle, very, very closely related. So it's in the same genus. We can distinguish them morphologically. We can distinguish males and females by turning them upside down and look at the last sternite. That's the last segment at the abdomen. It's a leaf-feeding beetle, both as larvae and as adult, and they like to aggregate. As I said, larvae and adults feed on water chestnut. Why do we think this is a great organism? Because this is what it does in China. So here you have a mat of a pond infested by water chestnut, and that doesn't look so good, even from the distance. Once you come in a little closer, you can actually see that these organisms basically move almost as a line from heavily defoliated areas in the upper left-hand corner to almost untouched leaves in the lower right-hand corner. And the insects that we released on purple loosestrife when they initially colonize a site, they do almost the same thing. So there may be aggregation pheromones that are involved in that. There may be a benefit in terms of avoiding predation or overcoming plant defenses, but these organisms aggregate and then move through an area and then leaving defoliated plants with no green leaf tissue behind. The floating petioles are still there. The plant may not be entirely dead, and there may be a mat of decomposing material, but you get light penetration again into the system. And so this is what they do in China. So it looks really, really promising in that way. I went to China in June 2016. And Dr. Ding at that time was working at the Wuhan Botanical Garden, and you can kind of see where that's located in central China. It's a little warmer than what we have here in central New York, but nevertheless, the beetles were abundant in the area. He worked at the Botanical Garden, and we could just get into a little vehicle, drive a mile, get out, and collect these insects in abundance there, and you can kind of see what that looks like on the left side. I then realized how specific these critters are. While this is not a native species, not even to China, that's water hyacinth floating in among all the water chestnut leaves that are heavily attacked by Galerucella birmanica. You can kind of see what we want. We want the target plant here, water chestnut, to be attacked and leave all other plant species unharmed. That's the idea about host specificity, and I'm going to go into some details in a moment. We needed to make sure that the organisms that we brought back into quarantine were really only Galerucella birmanica. So there was a lot of sorting, a lot of cleaning, a lot of new plant material that need to be procured that was clean of all other associated organisms, to make sure that all that we got into the US was Galerucella birmanica, plus some associated or attached leaves, so that they actually had something to feed. I went there myself because there is no Chinese carrier or no international airline that will transport living insects from China to the US. So I had a hand carry permit that allowed me then to go through the customs inspections and APHIS inspections to get back to the US, and the package can only be opened in quarantine. So there was a lot of sorting work. The collection was really, really easy and quick. And then there was a lot of extra work to make sure that we had clean vials with a little bit of a rosette in there. And here you can kind of see a little bit of what the system would look like. And then I went onto the plane with a box full of insects, and everything worked reasonably well until I arrived at midnight in the Cornell quarantine. And that's where other people come in. So you see Audrey Bo on the upper left hand, and Wade happily working with insects in the quarantine facility at Cornell. So life in quarantine, this is not our life in quarantine, but life of Galerucella birmanica in quarantine really is involving a whole bunch of things, including trying to understand how we can rear the organisms. You see in the upper left-hand corner, we have a whole bunch of, initially with buckets in a greenhouse that's set at summer temperatures. We need to feed the organisms. Then we initially attempted to rear them in petri dishes on whole rosettes, and we could build up the colony only to then suffer setbacks once we try to overwinter these organisms. And that's kind of the right-hand box that you see there, where we tried all kinds of different ways, from leaf litter to Trapa water chestnut nuts to Phragmites stems that we bundled so insects could come in and overwinter in places that may have the right moisture, that have the right temperature. They like to aggregate. In China, they may be sitting under rocks, but they go onshore. That's where they need to hang out until water chestnut comes back to the surface, which in the US, at least in our areas here, is not until early July. So at that time, there are some really warm temperatures, and the insects need to hang out and just survive, because water chestnut is the only thing that they can complete their life cycle on, with one exception that I'm going to go into in a moment. What happened to us over the years was that whenever we tried to do something, our winter was hitting us hard because we lost a whole bunch of the insects, from hundreds and hundreds that we tried to overwinter. So you can kind of see the timeline here, going from left to right. We did a whole bunch of things with no-choice testing. I'm going to talk to you about that, both in 2016 and 2017. And we gave them a cold period, so we didn't need to feed them over the winter, didn't need to raise water chestnut over the winter. That's what they naturally do. And then we took them out of the refrigerator at the quarantine and tried to restart the colony from few individuals. And so over time, our colony has dwindled a little bit for many different reasons that are not completely understood, but are not unusual in quarantine and captive rearing colonies. We were thinking of refreshing our colony in last year, then decided not to do it. We were thinking of refreshing our colony in 2019. Right now, we have a lot of trouble, or not we, but other people have a lot of trouble getting biological material out of China. The relationship between China and the US is not, let's call it, at its best right now. While that was not the result of biologists doing anything, nevertheless, we suffer from some of the political consequences. Dr. Ding is not sure that he would get an approval from Chinese authority to export these organisms. We had an approval by Chinese authorities basically saying Galerucella birmanica is not an endangered species. It's not used for anything in China, so we were free to export it in June 2016. Right now, that may not be quite as easy. We will see how that may occur in the future. What we decided to do instead is trying to rear the colony through the winter and improve and increase the numbers. That suffered some consequences or suffered some setbacks due to aphid infestations on water plants, due to poor plant quality. Wade seems to have found a way around that, and the colony is rebuilding as we speak. What that does, though, it creates other problems right now for us, which means we need to raise a whole bunch of water chestnut plants in quarantine again, because we obviously can't grow them outside right now. We can't purchase them anywhere, thankfully, and the nuts are under a layer of ice right now. So that's a challenge that we're dealing with, because they're very voracious feeders. Hopefully we can maintain the colony and do some of the experiments that we have not completed due to low numbers that we wanted to do in the fall of 2018. So we had a couple of little setbacks, nothing major, And we think we will be able to do everything in the spring of 2019 and then submit a tag petition to the authorities to ask for field release of Galerucella birmanica. So here's a little bit about the life cycle of Galerucella birmanica. It has an egg instar, it lasts a few days. Depending on temperatures, it has 3 larval instars that feed, and you can see as aggregations, these are black larvae similar to the water lily leaf beetle. I don't think you would be able to distinguish them, but we haven't tried, and because we didn't feel it is necessary at the present time. Then the pupae is kind of are these yellow silhouettes that look almost like adult beetles that you see in the upper 2 pictures that you have. So the life cycle is completed within less than a month for 1 generation. At least these are the results that we have in quarantine. That will differ a little bit depending on what the temperature is, and then you have new adults come in and repeat the cycle. So that's what we're dealing with in Wuhan. There may be 5, 6, 7 different generations. Under our climate conditions, we expect at least 2, maybe 3, depending on what the temperature does. If you go to the southern distribution of water chestnut in Maryland or Virginia, we're not quite sure what to expect there. The insects, if they get the petition, if we get the petition approved, that's what the insects will tell us what's possible. So you have a little bit of a life cycle very similar to the water lily leaf beetle. So I want to step back a little bit because it will become important in the end. Why we're doing biological control? We're doing biological control or any other management options because we think the problem is the plant that we have at hand here. Here are the negative impacts that are documented or anticipated or feared for water chestnut by covering water bodies, and the resulting impacts on economic aspects or biology conservation. So when we think about any management that we apply, we want a success. How can you define a success? We like to define it through demography and populations instead of individuals. And that will become clear in a moment what I'm talking about there. So in part, initially it needs the suppression of the target abundance and also its dispersal or spread. So that's a biological definition of that. Without suppressing of the populations of water chestnut, we may not get to an ecological effect. The insects may kill individuals, but that doesn't necessarily result in ecological effect. Again, defined through demography and populations, because what we ultimately want is that native species that are now currently suppressed, whether these are plants or whether these are fish or whether that's zooplankton, that they actually increase and have stable populations. And we want to avoid non-target effects, meaning by releasing biocontrol agents or doing anything else, chemical treatment or removal by hand, we want to not affect the species that we want to protect. So we call them non-targets. Yet we also know that by spraying or by doing mechanical removal, all kinds of other effects are there. Typically, they are not very well documented. Then there could be social or economic aspects that are part of a success. If people don't accept biocontrol as a method of choice, we're not going to have social acceptance. And what the economic impacts are for anglers or recreation, fishing, boating, whatever you have, that may also be important aspects of a success of a biocontrol program or any other management options. I want to note here that our biocontrol agents can only influence the biological success. So they can affect the way that Trapa grows, the way that Trapa increases or has decreases in populations. They cannot affect per se how other organisms are responding to that. So that's out of their realm. There may be, particular for terrestrial plants, there may be replanting efforts. I'm not sure that we can do that with aquatic plants, but that would be something that we can discuss. And I'm sorry, there's a note as a typo here. Biological success does not automatically result in ecological success. So we may reduce water chestnut populations, but water chestnut may not be the ultimate cause for the deterioration in ecological conditions. So if water chestnut disappears and hydrilla comes in, we may not have an ecological success of safeguarding native populations of fish or other organisms. Very often, lack of funding prevents a rigorous assessment of control programs, whether they are biological or not. So here, this is important because we need to assess what we do under those definitions of success. We can talk about that a little maybe at the end, if that is not quite clear, what I'm referring to here. So then if we think about that, what should be of concern as we're assessing whether our organisms are potent organisms to control the species and whether they have no non-target effects. Again, I want to focus on demography, and that includes both the target, here water chestnut, and non-targets, all native species. Selecting herbivores that cause heavy damage to target invasive plants but do not affect their demography are very poor choices. Biocontrol has made mistakes in the past. For example, they introduced a whole bunch of flower and seed feeders for species that were not seed limited. That includes a bunch of biennial plant species where 80–90% of the seeds were destroyed, but the populations were as abundant as they were before. So that's not a good choice of a biocontrol agent. We want to make sure that they are able to reduce populations, at least from where they're very, very abundant. If they occasionally feed on other species, or maybe even cause death of an individual of another species, but the populations of these other species are not being affected, that can be tolerated. That's kind of the definition of the Endangered Species Act, right? So an individual can be taken accidentally or otherwise, as long as the activities that you do not threaten the continued existence of the species. So there's a little bit of advanced thinking that we're proposing here, not only for water chestnut, but also other programs that I'm running, including on Phragmites. Not every nibble out of a plant will cause that plant to die or populations to disappear. Just think about the monarch butterfly or some other organisms that can defoliate their host plants without threatening their populations. So we wouldn't think about the monarch larvae being a threat to milkweeds, for example. And so we do the same argument here for host specificity testing of organisms, or for testing the efficacy of the organism to control a plant that we consider a problem species. So demography, in the way that we think about is, when we think about human populations, the WHO, the World Health Organization, is projecting how large human populations will be in the future based on information that they have about the age structure of human populations in different countries, ideas about the fecundity that differs between different populations and different countries, and other ecological or environmental factors. So there's a forecasting that people do with the information that they have from past behaviors of humans, fecundity, economic status, all kinds of other things. And we can do the very same thing with a little bit of math for plants or insects or whales, whatever you have. So we're thinking about the impact on future population sizes. And as I said, these can be ecological factors and it can be environmental factors. When we think about ecological factors, there are all kinds of interactions that may determine how large a population of a plant or any other organism may be. It could result interactions in terms of competition. There could be diseases, epidemics of dirt. There could be predation or herbivory in these. They can all affect the survival of individuals and the future population size. So these ecological interactions and some environmental factors, climate, rainfall, storms, all kinds of other things, affect individuals. And they alter both the survival, the growth of individuals, and the reproduction. So that's kind of in a nutshell what demography is. I hope it is clear to you what we're trying to talk about here, and I'm going to go into some more details in the following. So the initial idea is really assessing the impact to the target species here, water chestnut. The tool that we're using is demographic models because it allows us to quantify and upscale from individuals to population level effects. And what we do is we estimate something that's called the vital rates. And that's really the survival of individual life stages and to transition to the next life stage. So when we think about humans, you can think about the survival during pregnancy, of how many from conception to birth, how many individuals survive that, or after birth, how many babies will get to be teenagers, how many of the teenagers or young adults will actually reach reproductive age, and how many babies will they have. So these are different life stages, and the vital rates are the transition from one to the other. It's not always 100%, that's clear. But once we have those data, we can forecast the future population growth rates, and the mathematical symbol for that is lambda. So that's what you see in parentheses here. The big picture is that it provides a shared metric, lambda, for comparisons between different ecological conditions or management scenarios. So for humans, we can compare lambda between populations in Germany and the US, or Canada and South Africa. Because they will have different economic conditions, they have different population sizes, area, whatever else you have, different healthcare factors, but it provides a shared metric, which is lambda, population growth rates. And lambda can come in many different ways. When lambda is 1, when lambda is less than 1, we are seeing declining population. When lambda is larger than 1, we see increasing populations, and when lambda is 1, we have stable populations. How do we do that, or how did we try to do that? By establishing a whole bunch of little ponds, outdoor situations, because water chestnut we can obviously grow outside. And different populations may grow differently. There may be some taxonomic or genetic differences between the populations that we have. Some of those on the webinar may actually have collected nuts, rosettes, and send them to us. We appreciate that help. So we tested 120 different Trapa natans individuals from 4 populations over 2 years. We looked at how large they grew, how many nuts they made, what the overwintering success was of those, and then we could use those vital rates and survival and plug them into a model. And I'm not going to go into the math here to then look at what the effects of our herbivores were on these different vital rates. And here you have something how that would look like in a journal. Or in a formula. Again, I'm not going to go into the big aspects. SB stands for seed bank. S stands for seed. SM is small. LG is large because rosettes can be large and small. And you can go through this from fall to spring to summer back to fall. So basically, you have an annual situation here. And the different errors depict the transitions, and we can put in percentages of how many seeds would germinate from the seed bank and become small rosettes or large rosettes, or stay in the seed bank. So that's the first transition from up to low. How many of the small rosettes for Trapa would become large rosettes, or they may small. Some of the large rosettes may decline for whatever reason and become small rosettes. And in the end, they were producing some seeds, which will then go back into the seed bank. So we're looking at these. We have different groups of individuals that we follow, and we look at the survival, the germination, the growth to a small plant, the overwintering survival, and again, the number of seeds produced. It's reasonably easy to do that for water chestnut because it's an annual plant. It's not quite as complicated as long-lived individuals like whales or oak trees, for example. So when we did this, and these are outputs of a model here, so the upper 3 panels from left to right is the number of plants that would grow from a couple of individuals or one individuals under different scenarios. And you can see numbers can be very, very large. The different colors in the lines are just different populations, Maryland, New York, Virginia, or Rhode Island. Those are the ones that we had. But if you don't do anything, your populations will increase. And that's no surprise for those of you that have tried to manage water chestnut. We don't do anything, populations will increase. That it levels out at some point. These are the number of years at the bottom of those 3 upper level graphs. Water chestnut will reach the habitable environment. So if you have a pond, that pond will be filled. If you have a lake, water chestnut can grow only to a certain depth. That's why it's not forever growing. If we think about conservative biocontrol, the plants will still continue to grow there. The populations will expand. There are some interesting differences in the way that the different populations behave, but I'm not going to pay a lot of attention to this center graph. The one to the right is kind of where we see our Galerucella birmanica operating. It may not be as quickly as this is being depicted there because we have we're not entirely sure how long the seed bank actually lives. We think it's a very short-lived seed bank, and we are not sure how natural enemies or overwintering survival of our Galerucellas may affect that. But you can kind of see that over a reasonably short period of time, less than 10 years, maybe only 6 or 7, we will be able to suppress the populations of water chestnut effectively for all the different populations. And that largely happens to differences in growth rates. And we are now looking at the lower 3 hand panels there. What is depicted there is the growth rate lambda. And remember that 1 is the important aspect where a population is stable. And so you can see under the no action scenario, the growth rate will initially from just an individual or so will be very, very large. And then it goes down to 1. So why does it go down to 1? Because Trapa has reached its limits of which areas in a particular pond or lake it can occupy. So then the population will be stable, but it means that your lake is fully filled or your pond is fully filled. That's why it levels out at 1. It's stable. I'm going to go to the third panel, the lower right-hand level panel, and that's where we can kind of see once we release biocontrol agents. Early on, before the agents are very well established, you may get into a stable population for a couple of years, and then the population growth rates will decline. A declining population growth rate doesn't mean that Trapa is immediately disappearing, only that it doesn't grow or that the populations will shrink over time. It's not immediately disappearing. It will take some time for it to be reduced. If you want to more fully investigate what I'm talking about here, there's a paper that we just published in 2018 in Biocontrol. You have this here. You can look it up once this is archived. It's open access, so you don't need to pay. It's not behind a paywall. You can look at the arguments that we are having here for water chestnut in some more detail. So we think we have a very potent organism. This thing will not just eat water chestnut, but it will control water chestnut over large areas. And so we're very hopeful for that. So number one, do we have a potent organism? Yes. Will it do what we think it will do for water chestnut biocontrol? Yes, we think that's what's happening. Now, is it safe? So that's the next one that we're evaluating. And so these are now the risks to non-targets. I already talked about that we had a host specificity test plant list, 57 or so on there. So there's a hierarchy of different tests that we're doing. Basically, what host specificity testing does is establish what we call the fundamental host range. Given no choice or other circumstances, no predators out there, we are knowing pretty well which plants this organism can actually feed on and develop on. And you can kind of think about this as a series of filters. And that's what is depicted here. You start with a big pool of species, and you do no choice tests and use plant parts. Those that are still then being accepted for the organisms to oviposit on, to feed on, you go to multiple choice, where you still do plant parts. And it can be, these are very reductionist experiments, and they become more and more realistic as you go through. If you still have some organisms, usually there's a fewer and fewer and fewer that pass through these different filters, you do multiple choice with whole plants or field choice tests. And then you are left with some where you still scratch your head and said, is this thing safe? Is this plant okay? Will the critter eat it? Once I release it, then you do demography exercises to trying to understand what the effect would be on plant populations, not just whether an individual is nibbled at. So for water chestnut, in a typical way, this is what it looks like. Petri dishes where we allow adults to oviposit or larvae to feed. You have a long series of that. You need to procure those plants. That was a lot of work to trying to get to that. Here you see all kinds of pieces depending on the size of the plant. You do individual pieces or entire leaves. You let them feed for a while and see what the survival rate is and what the oviposition rates are. One of the difficulties that we encounter with Galerucella birmanica is that it likes to lay eggs on all kinds of things, including the plant tissues and the sides of little jars. You see a female ovipositing here on the bulrush, where it has never been found on, other than as a landing spot. So here you have a number of what we call false positives, but you need to test these species then even further. This is how a choice test would look like. Here we have a species that I will go into more details. Here we have a water chestnut leaves and a leaf in the lower left, and a water shield Brasenia schreberi on the upper right. And you can see these individuals are feeding on it. And so that wasn't a surprise. But here you have a choice test between the host and the non-host. And then we have 7 or 8 species, I think it's 7, where we had some oviposition, some larval feeding, that we then needed to go to a multiple choice test. So what you see here is the setup that we developed in quarantine, where we now have potted plants. We have water chestnut, and I don't think you can see that, or maybe it's in the foreground, the second from the right, and a whole bunch of other plants. There is Rosa palustris is in there, Decadon, water willow is in there, purple loosestrife is in there, and a couple of other species, Didiplis, that's in there, where these organisms were feeding, and we felt we needed to go through a multiple choice test. Now, the problem that we encountered here was that our colony was not flourishing as much as we thought it should or could. So we are not 100% sure about the test results here. And so Wade has some questions about reliability, validity of the tests that we're doing. And so we were thinking of repeating this in the spring with the colony that we hopefully grow from where we are at right now. So to get to this graphic again, what did we do? We had no choice where we did plant plants, as I said, 57 species that we cranked through the system. We did a couple multiple choice, but not really with plant parts. But we went to whole plants. I think the 8 is wrong. It has to be 7. The field choice test, we did some testing in China because they have some of the similar plant species in China. But we can't do this here in the US because we do not have the permit to do these testing in the field. So we're relying on tests in quarantine that are not always 100% predictive of what would be in the field. So we have one plant species where we feel we need to get to the demography exercises, and that's water shield Brasenia schreberi. From the early testing, from some field observations, we kind of knew that occasionally Galerucella birmanica will attack water shield when it occurs in close proximity. Water shield is native to the United States, Eastern Asia, Australia, and Africa, widespread. It's food in Asia as so many other plants. It's valuable for fish and wildlife in the US because the seeds are being eaten. So that's why that is there. When it has dense populations, it decreases dissolved oxygen in lakes and other water bodies. It's considered problematic in many regions in the US, and you can kind of see the distribution in the upper right. And in many places, you have extensive herbicide management to reduce the populations that then people feel they can't boat or swim in areas where you have expansive populations of Brasenia. And this is how it looks like in the Adirondacks. It's a funky plant. Wade tries to figure out all kinds of things about how it grows. What's the life history of the plant? Not much is known other than the plant just flowers, supposedly wind pollinated, is above the water surface for a day, then goes down below the water surface where the seed develops, and at some point the seed sinks. He has done all kinds of stuff to try to figure out what's going on with this plant. It has rhizome growth, it has all kinds of overwintering buds. At some point he may do this again and has some illumination to us of how this plant is actually behaving and organizing itself to have a successful life strategy that allows it to thrive in the US and many other places. I can't go into the details here, but it's important for us to then look at what life history transitions we actually need to go to. We did work in quarantine here, as you can kind of see here, where we look at the way that insects are choosing between water chestnut and Brasenia. Actually, the test here that's depicted is between Galerucella nymphaeae, not birmanica from China, but from the native water lily leaf beetle. So what we wanted to do is assessing the risks to non-targets. Remember, that's what we go on by. Brasenia is available in China. It's a native plant species there. The organisms are attacking it, and Dr. Ding did a field experiment where he actually established a pond with potted water chestnut and water shield plants there, and he released insects and allowed them to colonize what was happening, and he was censusing the number of individuals over time on water chestnut or Brasenia as he had these ponds there. And what you can see is, yes, there's an occasional adult that can be found on Brasenia, and there are occasional larvae that can be found on Brasenia, but they're always very, very heavily preferring water chestnut if you establish them in a pond like that. And similar situations are occurring in the ponds that are part of the Wuhan Botanical Garden that's focused on a lot of water plants. So we think this in the field, with giving adults a choice of where to do their oviposition, is looking really, really promising, that there will be no discernible impacts on water shield in the field by the organisms that we will be proposing for release. So some of the experiments that Wade is doing to figure out the life history goes back to some of the things that I talked about. I talked about the overwintering buds, which is like the third picture from the left in the upper row here. Wade will have more to say once he evaluates some of the data, once he has more information, maybe next fall. But there is a twist to the entire situation. The water lily leaf beetle that typically attacks water lilies has a whole bunch of different host races. It attacks loosestrife, for example, and it attacks water shield. There is supposedly a host race of Galerucella nymphaeae on Brasenia. It doesn't occur everywhere. I've seen it in the boundary waters attacking water shield. I've seen it in the Adirondacks. Wade has found a couple of ponds here. And this is what the water lily leaf beetle, at least the host race to water shield, does to water shield. And you can see there's heavy, heavy damage and yet water shield is still thriving in places where it's becoming so abundant that it's considered a weed that people want to manage with mechanical or herbicide. So this damage to water shield is so much more extensive than what we ever would expect from Galerucella birmanica. So, and water shield can tolerate this just fine. So we think the plant is safe from if Galerucella birmanica would ever attack it. But we're going to go through the demography exercise. So you saw this note, this demography diagram kind of on the left side. That's for water chestnut. So it's just here to depict what we're going to do. There will be different symbols in there. There will be a seed bank. There will be seed. There will be rosettes. We're not entirely sure how to do it yet because we have individual floating leaves that are attached to a rhizomatous plant. But it's a long-lived perennial. It has clonal reproduction. It has a reproduction by seed, and it has the major native herbivore. So how will this all fit together? I can't tell you that at the present time, but we will go through the exercises and do that. That's the setup that we're going to have for that, and as you've seen various pictures of that, it involves rows and rows and rows, hundreds of tests that we will do in the field where we can, and in quarantine where we have to. So this is a large summary, and this is an interesting picture. This is one of our undergrads, Alex Beck, as he tries to, with Wade, to get through what water chestnut looks like when you don't do anything. So the shirt, this I press on, is probably fundamental to what we need to do in terms of managing the species, but also understanding what does water chestnut actually do to the water bodies, to the organisms that are in these water bodies. Is it as detrimental as this looks like? And Wade will tell you much more about that at some point when he has finished his master's and then his PhD. There's some surprises that he will have for you. I'm not going to give them away. That's his story to be told at some point. So what do we have? We think we have a good organism here, Galerucella birmanica, strongly prefers Trapa, with major impacts on plant demography. That's what we want from a biocontrol agents. Some individuals can complete their life cycle on water shield, but even if you test the offspring afterwards, they still prefer water chestnut. It may spill over onto Brasenia from water chestnut in the field when plants are growing next to each other. But that damage is minor and of no apparent consequences to demography. But as I said, we will formally test that and then report that. The major herbivory by the native Galerucella nymphaeae appears no problem for Brasenia schreberi. This damage far exceeds what is expected from Galerucella birmanica. So we think what we should do, we think we have a perfectly safe organism that will occasionally nibble on water shield. There shouldn't be any ecological or economic consequences out of that. So I hope that the decision will be to go forward and release these organisms so that we can repeat what or have the impact that Galerucella birmanica has in China at some point in the US. In terms of a timeline, that's probably 2-3 years away, depending on how fast we can complete these tests and how fast the technical advisory group is working. That's what I have to say. With that, I'm ending my presentation, and I'm happy to answer all kinds of questions that will pop up. And I think Audrey will moderate that. Thank you very much.
Okay, thank you so much, Dr. Blossey. That was great, very informative. We already have some questions rolling in the chat box, so I'll start by reading those or reading them as they come in. If any members of the audience would like to submit questions, please do so. And as I mentioned at the beginning, we left a kind of a loose ending time. We may go over the 12 noon cutoff in order to address some of the questions more completely. So our first question is, what about fish predation on these organisms? There was great hope for biological control of Eurasian water milfoil with the moth and weevil, but that didn't fulfill the promise as planned.
So we assessed invertebrate predation on the native water lily leaf beetle, and there is. So we can have ladybugs eat the eggs. We can have water striders eat the larvae. you can kind of see that these larvae may not be able to complete development on one leaf. They need to go to the next one and pupate there. So there will be some mortality as they are swimming or trying to crawl from one place to the other. We are not sure about fish predation on these. They are not in the water per se, so the fish would need to come up to the surface and pluck them there. That's not unheard of for trout and other organisms, but I cannot quantify that at this time. Now, at the same time, these organisms doing this to China, where there's all kinds of fish in these waters. So we have anglers in the areas surrounding water trapa beds. They're fishing there. How our sunfish may respond to that, I cannot test that, or I cannot assess that at the present time.
Okay, our next question then is, so you are not concerned it could wipe out water shield after water chestnut is eliminated from the system?
No, not at all, because the survival rate of the, so again, think about it in terms of demography. While individuals of Galerucella birmanica are able to successfully complete their life cycle on water shield, they cannot build up populations. It's a death trap for them, because while individuals can feed and successfully develop, they cannot increase their populations. The mortality over time is larger than what would come up in terms of birth rates and others. The survival rates are so low, water shield by itself cannot maintain these populations. That's impossible. It's the same for the water lily leaf beetle. If you just have water chestnut, the water lily leaf beetle here will not be able to maintain populations just on water chestnut. The demographic consequences in terms of mortality of eggs or in particular of larvae are too high. They cannot maintain that without having access to water lilies. It'll never happen. And then in addition, water shield has a much, much larger distribution area in the US than water chestnut has. And the damage that it can tolerate from the native water lily leaf beetle is so large, it will suffer no demographic consequences. So the probability for that Galerucella birmanica would have negative impacts on populations of water shield, while not absolutely zero, extremely small.
Okay, thank you. The next question comes from Rob Williams. He would like to know, should birmanica be permitted? Is there a plan to raise bugs in sufficient numbers for additional releases?
Well, the plans are there in our heads. How are we going to do that is not entirely clear at the present time. As soon as we have a release permit, we can actually move things out of the quarantine and do mass production in common gardens. That's so much more successful in terms of population buildup. We may continue to rear some in greenhouses where we can potentially build up larger numbers. The way that I foresee that happening is that we would kind of wake them up from overwintering sooner, so we can get a generation or two in a greenhouse going for then appropriate field release by July, so that then they would have a couple of generations on water chestnut in the field in New York or wherever else we have. How are we going to deal with that? I'm not entirely sure, but yes, the plans are being formulated as we go forward. There will be some mass production, and we hope to have some collaborators in different places as well, where there's lots of water chestnut.
Okay, our next question is from Cynthia and Jeff. What life stages are G. birmanica using for overwintering? That is, are they overwintering as eggs, pupae, larvae, or adults?
Only as adults. So that's typical for most, I would say all Galerucella species, they overwinter as adults.
Okay, our next question is, you mentioned field releases in about 2 to 3 years. As a best guess, at what point do you think this will be a viable large-scale control method for use? Example, Seneca River.
The Seneca River may be one of the early release sites. So there's a little bit reading out of a crystal ball because I can't predict that. I can't predict how quickly these organisms will adapt to New York climate, how many generations they will have. But we need very few individuals, 50 to 100, I think, to establish an initial population if you do a release in a field cage, like say like a floating field cage or so. They aggregate too, so they like to find each other and hang out together. In the end, it will be similar to what we did with purple loosestrife. Probably an initial release will be 1,000, maybe larger if you have huge infestations, but we like to spread them out so that they can go about their lives in the different places that we have. If things work out okay with the permits and everything else, 5 years from now, we should be able to spread these things very rapidly through the area that Trapa occupies right now. Not every pond, but we can think about regions, whether these are prisms like in New York or watersheds or something. That should be possible because these organisms show explosive growth rates, and different from the Galerucellas on loosestrife, they have multiple generations. So that's really helpful here.
Great. Okay, our next question is from Cynthia S. She would like to know, once the leaves are destroyed, does that actually kill the plant and prevent it from setting seed?
The answer is yes. So it depends a little bit on when your organisms are attacking. So the picture that you still have up on screen shows that the organisms move from upper left to lower right. So if you have a rosette where the insects only get to in August, there may be some ripe seeds that have formed already. Those will still be set. But if you have defoliation, even I think we have some data in some earlier papers that Dr. Ding did, even 50% defoliation or so reduces seed output to 0. So you don't need to have the destruction that you see here on this site to affect reproduction. Yeah, that will prevent seed set very quickly.
Okay, we have a comment here from Wade Simmons, who's a graduate student that's working on biological control of water chestnut research together with Dr. Blossey. He mentions, and Wade, I'm going to make a correction here that someone below mentioned. I think you may have written this different than what you meant it. We also have not, so Wade says, we also have not found any habitats with both water shield and water chestnut. Let us know if you know of any. That would be a great field site for us to work in. So Wade, why don't you go ahead and type in the chat box your email so that anyone who has found a site like that and would like to reach out can reach out directly to you. Okay, and then we have one other question right now. Let's see. It's from Judy. To what extent can Galerucella impact other invertebrate species using aquatic plants where Trapa is occurring?
I would like to have a little bit more context for that, because what is happening here goes into many different levels, right? Because you have the stripping of the upper epidermis of water chestnut, and if the plants stick up, the lower epidermis. That increases the the ability of light to penetrate into the water column. That should encourage other macrophytes to grow. It would also encourage a lot of phytoplankton to grow. And what the insects are doing, they're fertilizing the plant column or the water column here, right? Because the poop that rains down is like you sprinkling fertilizer, which initially was taken up by water chestnut out of the sediments or out of the water column, and it becomes bioavailable for all kinds of organisms. initially plants, but then also zooplankton or fish or whatever. So what the effects would be on other invertebrates depends on what or other organisms depends a little bit on their trophic level and how other organisms are responding to that. We're trying to come up with assessment methods allowing us to evaluate that a little further. I cannot make predictions other than what we see early on would be a fertilizer effect from all the poop that rains down into the water, and the increased light penetration. So productivity of other organisms should increase. That should favor other invertebrates that may graze or sit on other plants. But there will be food web effects that at the present time, when you have dense water chestnut beds like this, is a little bit unpredictable in how that would shake out.
Okay, well, we've reached the end of our questions. If anyone has one last question you want to send in, please type quickly. If not, I'd like to, oh, we have one last question, and I guess we can make this our last one. Are there plans for seed bank analysis of substrate? That's from Michelle.
I'm not entirely sure that I understand what the question is, whether that goes after seed bank of water chestnut or seed bank of native species. I'm not entirely sure about that.
Michelle, would you like to type in any clarifications on that question? Well, we have not heard back, and so I'd like to.
Let me address that then, Carrie.
Okay.
Okay. So we have not thought about a formal analysis going out into the field beyond what we would do for demography analyses for water chestnut. What may be crucially important for those of you that are charged with managing water bodies, watersheds, or even larger regions is thinking about what should happen in your environment should water chestnut become under biological control. As I said before, our insects can only affect water chestnut. They are not able to then determine whether afterwards we have water lilies there or bulrushes or something else. If there are situations, other floating aquatic plants or submerged aquatic plants that you want in these systems, it would be up to you to determine whether they are present, whether that's in the seed bank, in the sediment seed bank, or whether they are present as individuals growing. And if they are not, it may be important for you to consider restoration efforts, either through seeding or through planting efforts. We typically don't do that for floating aquatic plants, as far as I know. We do it for some wetlands. We do it a lot for grasslands or maybe even some forests. But the Galerucellas, if they are released on water chestnut, may provide you with a window of opportunity where you can reset the system to something that's more ecologically or of higher ecological or conservation value. That's up to you to decide what you want in those places. And whether that's successful or not would require some trial and error approaches. But we missed a window of opportunity when we did releases of Galerucella for purple loosestrife. In some places that went really, really quick. And then in some instances, reed canary grass came in or invasive Phragmites came in, and not always did useful or rich plant communities develop after biocontrol of purple loosestrife. We missed a restoration opportunity there. So be prepared for that. Think about what you want into the systems and then potentially apply that once these insects become available, and trying to see whether you can work in concert with these insects to set the systems to a state that you appreciate more. That would be my recommendation.
Okay, we also had, and it seems like you addressed the question. We now have, the next question is from Cynthia and Jeff again. We now have hydrilla in the Connecticut River watershed. Do you think that the absence of the shade that water chestnut provides will increase growth of the hydrilla where both are present?
Anything that wants surface light would increase in growth rates once the dense canopy of water chestnut is gone. But we also see hydrilla growing under water chestnut. So we got unwelcome presence when we got water chestnut sent to us from Virginia and other places where hydrilla was part of that. So as I said, our biocontrol agents can only address the one plant species that they're able to feed and develop on en masse. They're not going to be able to give you a better ecological system if there are other invasive species in the waiting. That's the unfortunate effect. I can't solve that issue, and neither will be the Galerucellas.
So Barbara would also like to know if this also applies to Eurasian milfoil.
Potentially. Any plant that can grow below water chestnut and it's there, any plant that's in the water column can respond positively. That's why I'm encouraging people to think about how you want to restore systems. What do you want to put in its place? If you just allow anything that's floating around there to then go back into the systems where water chestnut is, you may get water milfoil, you may get hydrilla, you may get native potamogetans, you may get scirpus, you may get all kinds of things. I cannot predict that up front, but if you have water milfoil below water chestnut, water milfoil will respond positively if the water chestnut disappears. I think this is just a word on water milfoil, Eurasian water milfoil control. If you allow the systems to operate longer, water milfoil as an issue, particularly in larger lakes, will go away with the moth. The predation is happening on the native weevils. What's his name? Anyway, you know what I'm. Eurychiopsis is the native milfoil weevil. In part, people are not patient enough to allow the system to allow it to play out. And harvesting and herbiciding of aquatic macrophytes is not a solution for the long-term recovery of these lakes. That's just my little piece on